Ladies and gentlemen, thank you for standing by. Welcome to Pluristem's Investor and Analyst call discussing top-line results from its phase 3 study of muscle regeneration following hip fracture surgery. All participants are present in listen-only mode. A question and answer session will follow the formal presentation and will be accessible for analysts only by clicking the raising hand option on the bottom of the screen. Other participants, please submit your questions by clicking on the Q&A box. We will now present the forward-looking statement, which will also be displayed on our website once the presentation is uploaded. With us on the call are Mr. Yaki Yanai, Pluristem's CEO and President, Dr. NISSAN Halevi, Pluristem's Chief Medical Officer, and Professor Tobias Winkler from the Center for Musculoskeletal Surgery in Charité, Berlin. Professor Winkler is the principal investigator of our HipGen study and coordinator of an EU consortium conducting a multinational project on cell therapy for muscle trauma. And Franco Yehuda, Pluristem's Chief Financial Officer, will be joining us for the Q&A session. I would now like to hand over the call to Mr. Yaki Yanai. Mr. Yanai, would you like to begin?
Thank you. Good morning, everyone, and thank you for joining us for today's call. In the call today, we're going to review several items. Dr. NISSAN Halevi will present the phase 3 data, and Professor Tobias Winkler will discuss the clinical development, including our phase 2, which was the base for our phase 3 studies. I will conclude the discussion talking about next steps, and then we're going to open the call for Q&A session. Just to make sure that everybody is in line with all the clinical assets of Pluristem, we're going to discuss our muscle regeneration following hip fracture phase 3 study. And with that, I would like to hand it to Dr. NISSAN Halevi. Please.
Thank you, Yaki. And it's my pleasure to be sharing with you today the results of our phase 3 study. I remind you, these are the top line results. The study is still ongoing. Next slide, please. So just as an overview and reminder, this is from previously presented. This is a phase 3 study, randomized, double-blind, and placebo-controlled, which was conducted in 17 sites across the globe in the US, Europe, and Israel. It recruited 240 patients who came into the ER basically after a low energy trauma, which means they fell from a standing or sitting position with a fracture of their femur neck. They were then randomized within 48 hours of arrival into the ER and operated on for hemi or total hip arthroplasty or replacement of their hip joint. The randomization was a 1-to-1 ratio, either to 150 million cells of PLX-PAD or placebo, both obviously administered at the same time during surgery into the gluteus medius muscle. The primary endpoint of this study was short physical performance battery, or SPPB for short, and it was assessed at 26 weeks after the surgery and the administration of study drug. The safety is assessed at week 104. Next slide, please. Of this indication, we required our patients to be ambulatory or able to walk prior to the fracture, at least in their house, with or without any support. And of course, with this older age, it's understood that while this is a clinical study population and it is well controlled, there were patients with a lot of comorbidity that received a lot of concomitant medications when they entered the study. Next slide, please. The primary endpoint, which was a functional endpoint, as I said, SPPB or short physical performance battery at week 26, was not met. I just want to quickly explain what SPPB is. It is a series of physical tests that includes 3 domains, basically aimed at assessing lower extremity function and mobility. The first domain is a balance domain. Basically, patients are asked to stand and hold for at least 10 seconds the standing positions as you see to the right of the slide. So starting with normal standing, then a semi-tandem, and a tandem gait. When they are successful in completing this, or even if they are not, they go to the next part of the test, which is the 4-minute gait speed, basically measuring how quickly they can walk for 4 meters. When they complete this, the most challenging part of the test, and the last one, is the 5 chair stand test. Basically, participants are asked to cross their arms across their chest and rise from a sitting position on a chair to a standing position, and then sitting back down 5 times. And they have to do this as quickly as they possibly can. Obviously, a very challenging endpoint for such a frail elderly population. Next slide, please. So, despite our disappointment that the primary endpoint for the study was not met, we observed some very encouraging results for hip abduction strength. As you can see on the graph to the right, PLX-PAD is in red, placebo is in blue. We were able to observe, compared to a week 6, which is the earliest time point post-surgery that we can measure a hip abduction, a dramatic increase both at week 26, which is depicted on the graph as day 182, as well as week 52, which is the last point at the very right end of the graph. A meaningful increase in PLX-PAD strength of the hip, so hip abduction strength compared to placebo. Basically, while placebo-treated patients really had no meaningful change in their hip abduction strength from week 6 all the way to week 52. Patients treated with PLX-PAD showed a 2 kilogram increase at week 26 with a P of 0.047 and a 4 kilogram increase at week 52 with a P of 0.002. So very encouraging results. This is the results for the leg that underwent the operation, so the injured leg. On the next slide, I want to show you that even for the uninjured leg, the leg that was not operated on, and the contralateral side from the side that the drug was injected into, we also see a very similar picture. Starting at week 6, patients treated with placebo, again in blue on the graph to the right, don't seem to have any meaningful change in their hip abduction strength. Whereas patients treated with PLX-PAD at week 26 show a 1.8 kilogram difference with a P of 0.073. And at week 52, very similar to the injured leg, show a 4 kilogram difference with a P of 0.0046. The next slide shows the absolute difference in hip abduction strength between patients treated with PLX-PAD versus placebo at week 52, so a full year after the surgery and the administration of study drug, was 2.6 kilogram higher with a P of 0.051. This is for the injured leg. The uninjured leg showed a similar, albeit less substantial difference of 2.2 kilograms with a P of 0.1. Just as a reminder, the hip abduction for this study, as these patients are elderly and after a hip replacement surgery, was conducted in a supine position. Basically, the patients lay on their back and were asked to distance their leg with as much force as possible against the investigator, who then used a device called a dynamometer to measure the strength. Next slide. And the final thing that I wanted to share with you was that while the SPPB as a functional endpoint did not, the strength increase did not translate into the SPPB as a functional endpoint, we were able to see a very encouraging trend at a different functional endpoint that was measured, the 6-minute walk test at week 52. While we had a small number of observations for this, only 36 patients who completed this test at week 52 for the PLX-PAD group versus 45 in the placebo group, there was a 30-meter gain in the distance that the patients were able to walk during a 6-minute interval. This test is an acceptable functional endpoint in many studies, and it basically evaluates the global walking function, including pulmonary, cardiovascular, as well as musculoskeletal function. And with this, I'd like to invite Professor Tobias Winkler to discuss with you the results of the phase 1/2 and compare them with the results of the phase 3. Tobias?
Yeah, thank you very much for the handover, NISSAN. So I will, in the next few slides, lead you through our phase 1/2A study and talk a bit about the differences between these two studies and also the similarities. This was our first in human study that we conducted following a lot of preclinical work that we conducted here in Berlin at the Charité with the transplantation of, in the first time, autologous and then allogeneic cells, PLX-PAD cells, into musculature, and we always saw a very robust increase in muscle force. In this phase 1/2A study, we used in the human, to my mind, most standardized muscle trauma that we have. This is a transgluteal, a direct lateral approach in hip arthroplasty. We included 20 patients undergoing total hip arthroplasty because of an osteoarthritis. So bear in mind, this is not a hip fracture, but osteoarthritis patients, and we followed them 2 years with a lot of functional tests and also with a accompanying biomarker studies. Next slide, please. The results were quite promising. We saw an increase in gluteus medius strength. We injected 150 million cells. This was also the dose that we used in the phase 3 study, and also 300 million cells, which was not as successful as the 150 million cells, which led us to decide this as a dose for the phase 3. If you look at the left-hand diagram, you see that this pattern that you see with the increasing gluteus medius strength here in comparison to the placebo group, which is depicted in blue, is very similar to what we saw in the phase 3. So we saw an increase in gluteus medius force over time. At this, here we had a pre-operative also muscle force, which was then used as baseline. And this was then compared to 26 weeks. Very robust data, I have to say. And here also in the middle, we saw an increase in gluteus medius volume. We did MRIs with these patients, which we did not do in the current collective. We saw that we had an increase in muscle volume, which was also not, if we looked at the MRI pictures, due to a fatty degeneration or a fat increase, but really muscle substance, which with a significant increase in gluteus medius volume. Next slide, please. What was also very interesting was that we saw a significant reduction in post-operative stress-related immunological changes, as depicted here exemplarily for cellular and for a humoral parameter, the CD4 T cells and the plasma IL-10, which in the placebo group, which means without PLX-PAD treatment, saw a lot of stress-related reactions, which could be almost abolished by the 150 million groups, which was also part of the reason why we then went into this more challenged also patient collective in the phase 3 study. Next slide, please. If you now compare the two studies, then of course the phase 1/2 was not the multinational site, but the monocenter site. We did it at Charité. It was very, of course, in this way, very highly standardizable. Whereas in the phase 3, we had 17 sites with different surgeons and one approach. And so also in this perspective, of course, not as standardizable. The study population was an elective study population in comparison to the emergency arthroplasties. And also we had, as NISSAN already said, we had an increase in the mean age of more than 10 years in these patients, and also an increase, of course, in the comorbidity indexes that was really relevant here. The sample size, it was 20 patients in the phase 1/2 and 240 patients in the phase 3. And we used a single dose of 100 million cells versus placebo applied at the end of the surgery intramuscularly via 10 injections. The efficacy endpoint in the phase 1/2 I showed you was where the change in gluteus medius strength and volume at week 26, and the primary endpoint at the in the phase 3 was the SBBB that NISSAN already showed you in its complexity at week 26. Secondary endpoints were muscle strength and muscle mass and volume, and also hospitalization time and lower extremity measure as a patient-related outcome score. The study length is 26 weeks for efficacy in both, although we also have, of course, efficacy data on week 52 planned and also demonstrated as in the last slides. And we will follow up the patients in the phase 3 for 2 years for safety, which we also did in the phase 1/2 study. Next slide, please. So to sum it up, what you have seen here was a very, very robust proof that the PLX PAD cells can increase muscle strength. That can also be seen in such an unstandardizable patient population and multi-center study as in our phase 3 study. And so we are absolutely sure that the PLX PAD now trigger muscle regeneration and maintain it over time. What they also do is they obviously increase it over the time of 1 year. That also has been shown you here, and this confirms also our phase 2 results. I want to again imply that we have an older population, more comorbidities in this phase 3 study that also Dr. Halevi already declared, which surely also was a very, very demanding patient population for also during the corona pandemic, as you can imagine. But it was really successful in a way that we managed recruitment here, and we managed patient treatment and also the follow-up. Based on this data, I am very optimistic that we will be able to use the cells in a lot of potential indications in the future, namely all indications where you have muscle injuries as a patient, which can be sports injuries, it can be surgically related injuries, as we showed you in our studies here, which is the so-called iatrogenic muscle injuries or approach-related muscle injuries. And there is also the large group of traumatic muscle injuries that can be addressed by the treatment. Next slide, please. With that, I'll hand over to Mr. Yanai for the vision.
Thank you, Tobias, for this interesting presentation. And now I'd like to discuss our plans for next steps, how to move forward our muscle regeneration program. Basically, our plans are in 2 different directions that are both of them are very important. One of them, the regulation aspect, and the second one is pushing good forward with partnerships. Talking about regulation, I would like to give some background, which I think is important to understand the next steps and what we intend to do. Before launching of the phase 3 study, obviously we had several interactions with both FDA and EMA. Just to remind you, the PLX PAD was under the adaptive pathway in Europe. I think that both regulators agreed that we have showed a nice and strong phase 2 data showing the hip abduction and muscle volume. As you know, FDA wanted to see a functional endpoint in the phase 3. EMA were more open to discuss an endpoint like hip abduction, which is an endpoint that they were able to consider for registration. Eventually, and after a lot of consultation with our KOLs and of course with the regulatory advice, we thought that STPB will be the right endpoint based on the finding that we had for hip abduction. Just to give the right content, SPPB, we're talking about 5 or 6 years ago, it was like a rising star for companies that are trying to find an endpoint for hip abduction or muscle regeneration in general. And this endpoint was backed up by several several studies that were supposed to be initiated by major pharmaceutical companies like Novartis. And our goal and hope was that these companies will be able to demonstrate and eventually validate this endpoint, which was to date was not validated, the SPPB. Looking backwards, and today we are like 5 years from the day that we started this discussion and different companies launched studies, we do not see that none of the pharmaceutical companies were able to demonstrate any validation or confirmation for the SPPB as a functional endpoint. And of course, with that, we will seek for regulatory advice with the agencies in order to make sure that we would like to find a bridge between the very strong, solid, and robust data that we were able to repeat in the phase 2 and the phase 3, with to getting their advice, how we can translate that to functional endpoint. We see this gap, and we are committed to make sure that this product will be able to treat patients and to be able to regenerate and to increase the muscle force that is so needed for these indications. In addition to that, we will, and in the last year, we've been with quite a lot of discussions with potential strategics for this indication. I must say that we need to put this data, the outcome of the phase 3 in the right content. As far as we know, and what we've seen to date, we are looking at unique evidence that a drug that has the capability to significantly increase the muscle force with a single administration. And I think that all of us heard Professor Winkler talking about the importance that it will be for patients in a different traumatic muscle damages. And also in the last 24 hours since releasing the data, I think that the feedback that we get from the clinical centers that have been involved, that these physicians are quite excited, thinking of the opportunity to have a drug that will support muscle regeneration, muscle force, treating these patients. So I think that we have a unique finding here. And our main goal as a company is to find the right partnership in order to push it forward and getting the right regulatory advice. We do feel strongly that we have a strong evidence here, and we like to see how we translate that to continue development and getting this product to market, treating these patients. Next slide, please. Talking about partnerships, I do want to emphasize that July 22 is an important month for us at Pluristem. I do see that as the turning point of from here, we're gonna basically push forward the strategy that we announced a few months ago, where Pluristem will operate in 2 different arms. Of course, our pharmaceutical arms as well as the technological arm. Within the pharma, it is important for me to understand that we are making the adjustments to build a strategy that fits to Pluristem and our main core competitive advantage. While in the past we were relying on non-dilutive funding in order to push programs to phase 3, as we see here for the muscle regeneration. In the last year, we have adopted a strategy that is much more focused in partnership and collaboration with the pharmaceutical companies. We believe that this strategy will be an important part that each party brings his real competitive advantage to the table. We are a very technological company. We understand a lot about cell development and able to build a very sophisticated products and utilizing their main competitive advantage in bringing product to market. So our plans and goal is to enter to partnerships for this program as our phase 3, but also earlier in the process, in order to be able to push multiple programs in parallel. We are continuing to develop several very exciting early stage programs utilizing our knowledge and experience in developing advanced cell solutions. Today, we hold pools that we didn't have in the past, and we invest heavily in this very innovative and cutting-edge science, as well as understanding deeply the mechanism of action and the activity of the cells. We believe that it's an important part in order to pursue partnerships. And as I mentioned in the past through letters and calls, all of our future pipeline will be built on tailored and targeted products. In an ideal situation, we're going to have a product per indication. Today, we have the capabilities to modify the cells and to give them the competitive advantage for each and every of the product in order to be more effective in a specific indication that we are targeting. Just to mention that the PLXR18 that we published data a few months ago is our first modified and induced product, and we intend to do many more of this example using different techniques to alter the cells and their capabilities. And one of the main things that Pluristem is doing nowadays, we are focusing in several therapeutic areas. We believe that focus here is critical in order to be able to be an expert in a therapeutic area. And that's something that will allow Pluristem to be able to operate in more indication per therapeutic area. Next slide, please. And for the technological arm in general, I must say that we intend to continue to utilize our significant competitive advantage. Pluristem knows how to develop and manufacture cells in high quantities in a cost-effective manner. We're doing it in order to bring more value to our shareholders and to expand our proposition. We announced a few months ago the first collaboration, establishing a joint venture with Nuva, and we intend to continue to develop this technological arm for more partnerships that will be able to bring more business and more opportunities to Pluristem and its shareholders. Next slide, please. I would like to thank you for listening, and I would like to hand it to the operator.
Thank you. Ladies and gentlemen, at this time, we will begin the question and answer session. Analysts by clicking the raising hand option on the bottom of the screen. Other participants, please click on the Q&A box and submit your question. Before the question, please write your full name and company. Please stand by while we poll for your questions. The first question is from Chris from Jefferies. Please go ahead.
Hi, good morning, everybody, and thanks again for the presentation, and congrats on obviously the encouraging muscle strength data. I just had a couple questions. One was on the study conduct itself. What was the impact of things like physical therapy or exercise on the outcomes, and is that something that you plan to kind of evaluate as maybe an adjunctive to the PLX-PAD in the future? As it relates to that, as maybe slightly a little bit of a follow-up, you mentioned that there was a difference between the phase 1/2 and the phase 3 in a patient population, in as much as that the phase 1/2 was elective and the phase 3 was emergency procedure. So I guess maybe you could talk to what the motivational factors might be and perhaps the severity between those two specifically. And then the third question I would have is just maybe a simple housekeeping one. Just what would be the general timeline in terms of meeting with the regulators about these results? Thank you.
Thank you, Chris. So Professor Winkler will answer the first two questions about the different patient population and between the two studies. So please, Tobias, if you can go ahead with the answers.
Yeah, thank you very much. So the first question about physiotherapy is a very good one, I have to say, because obviously it has an influence, and we thought about that. This is why we recommended that our patients receive a standard physiotherapy, but we did not really interfere with this time. We did it in the phase 1/2 study. Because there we had a completely controlled patient population. Maybe I switch to the second question before I come back to the first now. These were people with osteoarthritis receiving hip arthroplasty, and hip arthroplasty is a highly elective surgical procedure where you have pain in your hip, and then you get an appointment, and after 2 months, depending on when you get an appointment, you get your arthroplasty and then you walk. And we could control all these patients. We sent them to the same rehabilitation institution. I trained the nurses there myself, and the physiotherapists, so I knew exactly what I would get back. In the case of the multinational, multi-center study, we had to deal with different physiotherapies in the US, in Israel, in Germany, and we just could give hints or tips concerning a standardized physiotherapy, but we were not able to control it. And what we have not done yet was that we did not evaluate the extent of physiotherapy or the extent or the type of the exercises on the outcomes. But I have to say that we had a very good randomization procedure, and this is what we thought that the randomization procedure would help us there. I hope this answers your questions.
Okay, Tobias, and to add to that, to the third question regarding the timelines, meeting the regulators. So obviously, we need to do a very good prep in order to provide a good arguments and a good position here. And that's something that we started to work from the moment that we saw the data. Assuming that's going to take us a couple of months, we hope that we're going to see the meeting and we're going to apply for the meeting immediately afterwards. So in the best case scenario, we'd like to see it by the end of this year or beginning of next year, but it's really need to be when we have a very good position, prepared everything, and we are confident with both of them. Just as in the brackets, I hope that's going to end soon, but I guess that as all of you know, there are some delays now with the FDA meetings because they're still on the COVID charter. So that's something that we do not factor right now, and I hope that it will not interfere with the guidelines from timelines that I just gave.
Thank you, Chris. The next question we have is from RK from HCW. Please go ahead.
Thank you. This is RK from HCW. Three questions from me. So on the first study, which Dr. Halevi talked about, I'm trying to figure out what the reason was that you had a smaller number of patients in the 6-minute walk part of the study. And then, were there any other tests for functional measures performed in the phase 1/2 study that Professor Winkler was talking about? And if SBBP is not really a validated endpoint, what makes FDA comfortable to use it as an endpoint for regulatory approval? And are there any other functional measures that FDA would accept other than the SBBP? Thank you.
Thank you, RK, for the question. NISSAN, please go ahead.
Thank you very much. Very, very good question. So first of all, as to the amount of missing information, obviously, you understand with this old population during the pandemic, this study was started towards the end of 2018. So many of our study visits actually occurred during the time of the pandemic. So obviously, evaluations like the 6-minute walk are very problematic to complete at home visits, where we were able to actually obtain a lot of other data and were implemented in this study. And we do realize, of course, that this was something that hindered us being able to collect this data. And it's something that we're evaluating deeply and trying to understand the impact on that, on the trend that we were observing. As to SPPB as a functional endpoint, agreed, it is not a validated endpoint. And as Yaki mentioned, at the time this study was conceived, there were several companies, including large companies like Novartis, who went out after this endpoint in several large studies, and unfortunately all of them had failed. Part of the, work that these companies, as well as us, were intended to do was try and within our study, try to validate this endpoint. If there are other functional endpoints included in this study, on addition to the 6-minute walk, we also have some questionnaire data and quality of life data that is also considered to be, somewhat functional or interesting or clinically meaningful in the FDA's eyes. And as to, sorry, I'm blanking on, the last question was for the phase 1/2 study, and did that include any functional measures? And I believe it did not. Professor Winkler, please correct me if I'm wrong.
No, that's correct. Not more than I showed you.
Thank you, RK. The next question we have is from Laura from Alliance Global Partners. Please go ahead.
Hello, this is Laura Serial covering for Jim Malloy from AGP. Thank you for taking our questions. So for the next steps for this program, considering the trial did not reach the primary endpoint of the SPPB, will you possibly revisit this endpoint in potential future trials, or what alternate endpoints might be utilized instead of this endpoint? And regarding the partnerships for this PLX-PAD program, what are the current levels of interest that are you seeing for this program, or what levels of interest might you anticipate for this program? And overall, what is the realistic timeline for a potential signing of a partner to continue the development of this program? Thank you.
Thank you. So NISSAN, please, NISSAN will refer to the first part and I will refer to the second part, or to the second question. Please, NISSAN.
Yeah, thank you for your question, Laura. I think considering SPPB in a future study is something we're not likely to do. As I said, we're still looking into this data. This is obviously only the primary efficacy phrase. But so far, we've not seen anything that would encourage us to use this endpoint again. From all the data that we've collected in this study, and I have to say, there's still a plethora of data that we need to explore. The 6-minute walk test seems to be the most promising for an alternative functional endpoint. Of course, all of this will be the result of regulatory discussions when we get to that.
Thank you. And for discussion of next step and potential partnership for this program, we do see a good level of interest from several parties in a product that will be able to support muscle regeneration. Think of the opportunity in how many indications we can actually tackle with an agent that do not require the compliance of patients for physiotherapy or whatnot, so it can be applied with a simple administration of a product, of a drug. So we see the interest that is there. Obviously, we would like to meet the potential partners, seeing the data. Today, we have a good, deep understanding of the mechanism of action that is able to support from a mechanism of action to preclinical phase 1/2 and a phase 3 that we are able to show the correlation. Obviously, as I mentioned, that's something that we need to make sure that we are providing a good outcome from a regulatory point of view. But overall, we feel optimistic about this outcome and the importance for that for the patients, and coming with a novel product that as of today, there is no drug or any other things in the market that physician can offer to the patients as a therapy to support muscle regeneration. Regarding timelines, I usually, and I think it's not very wise to commit on timelines for partnerships. As I mentioned from yesterday, we are working with all the parties that we have engaged in the last 6 months. We're going to present the data in the coming month, and we'll be more than happy to announce for any progress in the regulatory aspect as well as in the business development side.
Thank you, Laura. The next question we have is from Matt from Ladenburg. Please go ahead.
Hi, good morning. Thanks for taking the questions. I guess first, just wanted to dig into the primary endpoint, although you missed it. Were there any trends that you saw in that endpoint, and were the results or that miss of that endpoint specifically driven by any one of the 3 domains? Did you see any signal in any of the 3 domains that you measured there in the SPVB?
Thank you, Matt, for the question. At this point, since we are reading the primary endpoint, we are referring to it as a whole, and we are not breaking it out to the different elements as it's described during the conversation. And we do prefer to refer to mainly to think that we did see a significant trend or a statistically significant difference in order to make the observation. There is still a lot of data to analyze, and we'll be able to break it by the 3 elements, and we'll be able to share this information in later stage. I must say that in general, we are not in a position to be able to break SPPB to any part of it as a functional endpoint. Therefore, we would like to refer it as a whole. But there is a lot of work to do inside this functional endpoint, and as we said, to validate additional functional endpoint that will be acceptable for a product like that. I hope it answered the question, even though not directly.
Thank you, Matt. Before I ask Mr. Yanai to go ahead with his closing statement, I would like to remind participants that a recording of this call will be archived and available on Pluristem's website, pluristem.com, beginning tomorrow. Mr. Yanai, would you like to make your concluding statement?
Yes, thank you very much for attending this call. This is a phase 3 study with, I think, quite unique outcome. As I mentioned in our statement, we are very pleased to see that we have repeated the outcome from the phase 2, while in the same level, I'm disappointed that it wasn't translated to a good outcome of the primary endpoint. I do want to assure you that me personally and the entire Pluristem, we are committed to push forward this program through a regulatory and partnership involvement. And we are doing it because we do feel that we have something special or something unique that we're able to present here. And our patients definitely worthwhile that we're going to make the efforts to make this product available for them. So thank you all for participating, and we'll keep you posted.
Thank you, Mr. Yanai. This concludes Pluristem's call. Thank you all for your participation. You may now go ahead and disconnect.